The video today is going to cover chapter 3, the protein section of chapter 3. As we go into enzymes, we want to cover proteins a little more and what that's going to accomplish. So, chapter 3, proteins. So, if you notice a couple of things here real quick. So, proteins, if you're following the book, so chapter 3, section 12, proteins are literally one of the most important molecules. They're all important, but proteins are involved in nearly every dynamic function in the body that has any kind of diversity and everything. So just actually a short list of protein functions. So they act as an enzyme. This we're going to hit up a little more importantly here as we go into the lecture tomorrow. There's transport proteins, so we've talked about those and how they move things in and out. Defensive proteins, so your antibodies that fight off bacteria and diseases. Signal proteins, so hormones that cause your body to react to stimuli. Receptor proteins, so these are proteins that basically receive signals. They allow for molecules to be recognized. Contractile proteins, so these are found in muscles, so it's proteins that allow your muscles to contract and relax. Structural proteins, they build your body. Storage proteins, they store things. Literally, if your body does it, there's a protein involved. Also, we don't think about this, but the proteins are the molecule that give you your hair color, your eye color, your skin color, everything about who you are. That's super important in that process. So proteins are composed of differing arrangements of a common set of only 20 amino acids. So just to remind ourselves, so protein is the macromolecule here. So that's what we call the polymer. And the amino acid is the monomer of that. And so super key right here. So if you don't pick up anything else, the function is dependent upon the shape. So the function is very dependent upon the shape. And so that's going to be important in proteins, but in enzymes as we talk about that. And then the process of denaturization. So if you change the shape, you lose function. So basically remember here that that shape equals function. So that's kind of the key thing to remember as we go through there and work our way through. So just a general picture of what we're looking at here, a lot of different structures. We're going to talk a little more about these structures as we go a little deeper. This is kind of a ribbon model. This is the space-filling model, so you can actually look at the different atoms. If you use the model kit in chemistry, you'll probably recognize some of this. So this is just, it looks like a big giant glob, to be honest with you. So proteins, the monomer, the amino acid, contains basically 4 structures. We're going to talk about those. So we've got the amino group. We've got what's called a carboxyl group. Both of these groups are basically what we call a functional group. And much like the name says, a functional group has to do with function. So it kind of controls a little bit of the function. And then you've got an R group at the base. The R group, so this is just a variable. The variable is, like in math, just a placeholder. There's 20 different R's that can go in there, each with a very specific property of what it's doing. So to link our monomers together, it's through a process of a dehydration reaction. So when you dehydrate, you basically lose water, right? So you lose some H2O. So H2O is going to remove. And so if we look at how we lose H2O, we're going to lose an OH group. So that's what's called a hydroxyl group. And we're going to leave a hydrogen ion as well. So H2O, so there are two hydrogen or oxygen. And then those functional groups, the carboxyl group right here, so our carboxyl group, and then our amino group are going to join together, and you're going to form a peptide bond. So we'll see that in a minute. So that's what that region is actually called, is a peptide bond. With that being said, you'll also sometimes hear as a protein is called a poly means many, a polypeptide. So you'll hear it called as a polypeptide. And so that's what it says down here. So it creates that chain of where that's going to be. So this is the basic structure. Something you're probably going to be asked to do is kind of recognize this structure. So here's an amino group. An amino group consists of a nitrogen and two hydrogens. And then we got a carboxyl group. So we've got a carbon. We're going to have a hydroxyl. So this is actually, this by itself is called a hydroxyl group. Hydroxyl. And then this is a double bonded oxygen. So this whole thing is our carboxyl. The hydrogen just kind of floats around up here. And then this is your R group. So depending on what that is, remember there's about 20 different one of those that'll pop up that you can kind of see. So just for an example of what that looks like here. So there's three right here. So leucine, so again, if you see, here's your amino group, your carboxyl groups there. I'm going to just kind of skip over here, so same thing. Amino group, carboxyl group, amino group, carboxyl group, so they're all kind of there. We've got the only difference here is the R group. So this is a hydrophobic, so hydro, water, phobic, fear. So this one does not like water. Hydrophilic, philic is loving. So again, these like water. So as you can see, these three, and they'll react differently depending on the situation that they're in. So this little picture here shows what's going on. So here's our OH, our hydroxyl group. Here's our hydrogen. So that's going to make water. And as we go through our dehydration reaction right here, we're going to lose our water. So lose water. And we're going to form that peptide bond right there between the carbon and the nitrogen. And so then di is 2, so that's a dipeptide because there's 2 of them there, but a polypeptide is many. So protein, one of the key things to know about proteins is that they have 4 levels of structure. And again, as we've said, structure right here determines function. So it determines the function of that protein. So this is just kind of a brief overview. We're going to hit each one a little more detail. So protein's primary is its first structure, is just the sequence of amino acids. And so remember, there's 20 different ones, and so depending on the sequence you put those in, that's your primary structure. The secondary structure, so after we build this, the protein has 2 options to do. It can either do a coiling or it can do a folding, and we'll talk about what those are. And the key there is something to remember, and I'm going to highlight this because that's going to be easier. I think I can highlight. Is that a highlight? Yeah, okay. Is that it is stabilized by hydrogen bonds. So that's key to remember there, is that it's stabilized by hydrogen bonds. The tertiary structure, this is kind of the big one. This is when we talk about structure, it's tertiary. It's the overall 3-dimensional shape. So after it lines up, it coils and folds a little, this is the big guy here. And the thing to remember there, much like what we just saw here, is this is a result of R group interaction. So the R groups are going to interact at that point, and we're going to see that work its way through. And then finally, there could be a quaternary structure. That's its fourth level, and that's just where you have multiple polypeptides that come together to form an even bigger complex for various reasons and why. So let's look at each one kind of individually as we go through. So here's kind of the primary structure. So again, if you notice, it's just a long chain. So we have our single amino acid, second amino acid, third amino acid, so dot, 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 dot. So this order is super important. In our module 3, we're going to talk about the order here and how important that is. So right now, just kind of have faith and trust me that it's important to have that process, that order there. So the secondary structure, you've got two options here to know. So the first one is going to be an alpha helix, so that's the twist. And then you have what's called a beta pleated sheet. That is where it just kind of folds. And remember, super key important item there is hydrogen bonds are what control that. We've talked in our water unit. This is a weak bond, so obviously it's only kind of a holding place to kind of make some structure happen. Then we go into our tertiary structure. So now what happens is it starts folding even more and taking on a very unique shape that's very particular. So it's got to have a very particular shape. So this divot right here is important. This knot right here is important. And again, it's all about the interaction of our R groups. There is the key to that process. And then we go into our quaternary structure. So polypeptides are associated into a functional protein. So not every protein has a quaternary structure, but see, we can look here and I would count that there would be 1, 2, 3, probably 4 different tertiary structures there to make one molecule at that point. And so this picture right here just kind of puts it all together. So primary, secondary, tertiary, and quaternary structure as we go through. And these videos would play, but they're embedded, so they're not going to play for us at the moment. So the key things to take away from proteins, let me go back to the overall picture here. So the key thing to remember is proteins really are responsible for a lot of aspects of our body, of our structure, of our function. They are the sequence determines the shape, that determines the function. So that's a good way to think about all the linkage between that. And then they're put together through a dehydration reaction where you lose water. And then the primary structure, the secondary structure, the tertiary structure, and quaternary structure. So if you know that kind of gist, then you will be prepared to talk about enzymes and to answer any questions about proteins.